Good day. The lecture for this hour is on the physiology of the retina and the vitreous. Okay, so the objectives of this lecture are to know the physiologic function of various retinal elements, to understand the physiologic and biochemical process of vision, to know the functions of the vitreous, and to know the relationship of the vitreous and retina in health and disease. Now, this is going to be the outline of my lecture. Okay, so first we discuss the retina and then the vitreous. First, our retina. So the retina is a complex and highly organized light-sensitive structure of the eye. It transforms light energy into electrical signals, which is then transformed to neural signals for interpretation by our brain. To understand phototransduction, we must first be familiar with the functional organization of the retina. The retina is divided into a neurosensory retina and the retinal pigment epithelium. The neurosensory retina is further divided into its nuclear layers containing the nucleus of neuronal cells, the plexiform layers where axons and dendrites of these cells interdigitate, the photoreceptor layer containing the inner and outer segments of the photoreceptor cells, and the nerve fiber layer, which is made up of the axons of the ganglion cells that continue on as fibers of the optic nerve. So if you look at this picture, you see the neurosensory retina and the retinal pigment epithelium. The retinal neuronal chain is a 3-neuron chain from light source all the way to the brain. As I have mentioned previously, we have the nuclear layers, meaning the outer nuclear layer, which is where nucleus of the photoreceptors are found, the inner nuclear layer, where the bipolar, horizontal, and American cell nucleus are found, and the ganglion cell layer, where the ganglion cell nucleus is located. As I also said previously, the plexiform layers are where the synapses between the cells are found. The rods and cones are the photoreceptors involved in vision. They have the same basic structure, but differ in amount, location, and functional characteristics. In terms of number, there are approximately 120 million rods in our eye versus only 6 million cones. The rods are responsible for vision in low light conditions since they are very highly sensitive to light. Rod-mediated vision, have lower acuity and is achromatic, having only one type of photosensitive pigment. In contrast, cones are responsible for photopic vision. It is less sensitive to light but give better or higher visual acuity. They have 3 types of photopigments which allow for color discrimination. Rods and cones are found in the outermost layer of the neural retina. It is made up of an inner and outer segment, the nucleus, and its synaptic vesicle. Their cell nucleus is found in the outer nuclear layer. The outer segment of the photoreceptors are modified cilia containing disc filled with the protein opsin, the molecule that absorbs photons or photons of light. It also contains voltage-gated sodium channels. Opsin contains a pigment called retinal. The combination in rods is called rhodopsin, and in cones called photopsin or iodopsin. The different classes of photopsin in cones react differently to wavelengths of light, allowing for color discrimination. So in the unstimulated state or in the dark, the retinal bound to the opsin is in its 11-cis retinal form. Cyclic nucleotide-gated channels in the outer segments are open because they are bound, CGMP are bound to them. Positively charged ions, sodium, enters the photoreceptor, depolarizing it. Depolarization of the cell membrane opens voltage-gated channels of the calcium of voltage-gated calcium channels. An increase in intracellular concentration of calcium causes vesicles containing glutamate to merge with the cell membrane, releasing glutamate into the synaptic cleft. In the presence of light, 11-cis-retinol is converted to the all-trans retinol form. Opsin is activated to transducin, and sodium and calcium channels are closed, causing hyperpolarization of the cells, and this stops the release of glutamate. So this is a summary of the phototransduction pathway. So we'll go by it segment by segment. So in the first part, when light hits a photoreceptor pigment, opsin, the retinol bound to it is converted from 11-cis-retinol to the 11-trans form. Opsin undergoes conformational changes and activates a G protein, the transducin. Transducin in turn, activates cyclic GMP specific phosphodiesterase, which causes hydrolysis of cGMP to 5' GMP. The decrease in cyclic GMP causes closure of the sodium channels. So the closure of the sodium channel causes hyperpolarization of the cells. With the ongoing outward non-gated potassium-selected channel, this is ongoing, okay, across the cell membrane, this cell membrane becomes negatively charged. The now negatively charged cell membrane causes calcium channels to close, which leads to an inhibition of the release of the neurotransmitter glutamate. In the deactivation phase, or the so-called recovery phase of the cycle, rhodopsin or photopsin kinase phosphorylates the light-activated opsin, which binds to aristin and prevents activation of transducin and the subsequent steps of phototransduction. Hydrolysis of cyclic GMP is slowed down, allowing cyclic GMP channels to open. Thus, a new cycle of signaling can begin again. ATP provided by the inner segment powers the sodium-potassium pump, and this pump is necessary to reset the initial state of the outer segment by taking the sodium that are entering the cells and pumping them back out. Now, depending on the nature of the receptors in the bipolar cell, so we're already on the next part of the neuronal chain, it will either be hyperpolarized or depolarized with the presence of glutamate. When glutamate binds to an ionotropic receptor, the bipolar cell will depolarize. When it binds with a metabotropic receptor, the bipolar cell will hyperpolarize. So with light, there is hyperpolarization of the photoreceptor cells, which inhibits glutamate release. This will cause depolarization of the on-center bipolar cell and hyperpolarization of the cone off-center bipolar cell. On the other hand, when the photoreceptor is depolarized in the dark, glutamate is released, causing hyperpolarization of the on-center bipolar cell and depolarization of the off-center bipolar cell. In essence, this characteristic of the bipolar cell also allows one population of bipolar cells to get excited by light and another gets inhibited by it, even though all the photoreceptor shows the same response to light. So ganglion cells are the final retinal element in the pathway from the retina to the brain. It synapses with a bipolar cell at the inner plexiform layer. An off-ganglion cell synapses with an off-bipolar cell and produces an action potential when the off-bipolar cell depolarizes when light is on. In contrast, an ongoing an on-ganglion cell that synapses with an on-bipolar cell reduces its production of action potential when the on-bipolar cell is hyperpolarized when light is on. So the axons of these ganglion cells are what continues on as the optic nerve. So aside from the vertical neuronal transmission, we also have lateral connections within the retinal neuronal layers. The amacrine and horizontal cell provide lateral connection, which is important in adaptation and amplification of signals. So we have the horizontal cells synapsing with the photoreceptor cells in the outer plexiform layer. This is important in dark and light adaptation and in contrast sensitivity. These cells help integrate and regulate inputs from multiple photoreceptor cells and provides negative feedback to other photoreceptor cells. Amacrine cells synapse with bipolar and ganglion cells at the inner plexiform layer. They are responsible for movement sensitivity and enhances center surround effects in ganglion cell receptive fields. They connect rod bipolar cells to cone bipolar cells, allowing the ganglion cell to respond to the entire range of light levels. So aside from the neuronal cells, the retina also has a structural cell element. The Mueller cell is a glial component of the retina. Its food processes form the interlimiting membrane, and it also with the junctional complexes with the inner segment of the rods and cone form the external limiting membrane. The Mueller cells provide structural and functional stability to the retinal cells. So they uptake excess neurotransmitter, they remove debris, they regulate potassium levels, they store glycogen, they function as electrical insulator or receptors for receptors in neurons, and provide mechanical support to the cells. So all these neuronal cells that I've discussed are all found in the neurosensory retina. So now let's go to another retinal layer, which is the retinal pigment epithelium, which is the outermost layer of the retina. This layer is responsible for storage and conversion of vitamin A for synthesis of rhodopsin or photopsin. It is responsible for production of glycosaminoglycans that envelopes the photoreceptors. It has phagocytosis and helps degrade shed lamellar disc of the photoreceptors. Helps regenerate bleach photopigments. It also functions by absorbing scattered light and pigment screening from outer segment melanosomes, promotion of retinal adhesion and fluid transfer from the subretinal space, forming the outer blood retinal barrier through its tight junctions. So all in all, the retinal pigment epithelium is very important to photoreceptor health. The RPE is important in the visual cycle due to its role in the vitamin A cycle and in regeneration of bleach photopigments. Vitamin A in the form of beta-carotene in food is absorbed in the gut and it's split into 2 all-trans retinol, which is stored in the liver. The liver secretes all-trans retinol, which combines with serum retinal binding protein and is carried to the choriocapillaries. It reaches the RPE base, and inside the RPE, all-trans retinol is isomerized to 11-cis retinol and oxidized to 11-cis retinal. 11-cis retinal, which binds with interstitial binding protein, enters the outer segment of the photoreceptor cells where it is bound to opsin. So when light cleaves the bond of rhodopsin, 11-cis retinal transforms to all-trans retinal, which converts back to all-trans retinol which is transported to the RPE, which is then isomerized to 11-cis retinol, completing the cycle. So the RPE also has phagocytosis function. This located in the outer segment, which is enveloped by the microvilli of the RPE. So the RPE ingest and digest this oldest. So they do recycling and also they clear the debris. Dysfunction of this role causes accumulation of incomplete digested debris, which causes RPE stress. This is sometimes why we see drusen in age-related macular degeneration. The RPE also has a pigment screening function through its melanosomes. These are found in the microvilli at the apex of the RPE, which surrounds the outer segment of the rods and cones. They minimize scattering of light from one receptor to another, absorbs light energy like in photocoagulation, and it also has some affinity to certain drugs. The RPE is instrumental in maintaining retinal attachment. Passive forces as well as active pump system keep the neurosensory retina adherent to the RPE and keeps the potential space between them dry. So in this picture, we see a break or a breakdown in RPE function, which can cause fluid to accumulate between the neurosensory retina and the RPE. This causes a loss of contact between the photoreceptor cells and the RPE, which will affect the phototransduction cascade. So we now go to the vitreous. So the vitreous is a clear hydrogel occupying about 80% of the volume of the globe. It provides semi-solid support for the retina with firm attachment to surrounding structures. It is optically clear, allowing transmission of light. And metabolic pathways for it has metabolic pathways for nutrients to reach the lens and the retina. So the vitreous is 98 to 99% water. It also contains collagen and glycosaminoglycans. The index of refraction is similar to aqueous. The volume is approximately 4 milliliters with a weight of 4 grams, and the pH is 7.5. Generally, it is spherical with a depression at the surface called the patellar fossa. The vitreous can be divided into two zones. You have the cortical vitreous, which is near your retina, and the medullary vitreous. So the vitreous cortex is more condensed and it's fibrillar. It presents only around 2% of the total vitreous volume. It is in contact with the ILM or in the internal limiting membrane and is involved in production of hyaluronic acid. It is the metabolic center of the vitreous body containing vitreous cells, the fibrocytes, and hyalurocytes. The medullary vitreous, however, is essentially cell-free. It exists either in gel or liquid form depending on age, refraction, and condition of the eye. It has lower amount of fibrils, and liquefaction or syneresis usually starts in the medullary vitreous. So the vitreous is bounded anteriorly, has an anterior boundary and a posterior boundary. So the anterior hyaloid contains the greater density of collagen fibril. Superficial fibril runs parallel to the surface and is in contact with the ciliary process, lens zonule, and posterior lens capsule. The posterior hyaloid, however, is a condensation of vitreous fibril on the retinal surface. So the posterior hyaloid is only evident when you have vitreous, when the vitreous detaches from the retina. This is where we sometimes see accumulation of blood as the vitreous contracts. The vitreous is attached to the retina at the following areas. No? You have the vitreous base anteriorly, posteriorly at the disc margin. In between, we have it's attached to the fovea, major retinal vessels, and it may be attached to certain abnormal structures like lattice degeneration, chorioretinal scars, or even in photocoagulation scars. So the vitreous is firmly adherent to the retina at the vitreous base. It is approximately 2 to 6 millimeter wide, straddling the ora serrata. So the anterior margin is 1.5 to 2 millimeters anterior to the ora, or 5 millimeters posterior to the limbus, and its posterior margin is 1.8 millimeters posterior to the ora temporally and 3 millimeter posterior to the ora nasally. Another area where there is firm attachment of the vitreous is at the optic nerve head. Here, there is intermingling of the vitreous fiber with the basement membrane of the mular cells. In complete posterior vitreous detachment, when the vitreous separates from the optic disc, a ring of fibrous tissue is seen, known as the Weiss ring. The vitreous gives structural support and volume to the eye. It is a shock absorber, has scavenger function, a metabolic repository, and has optical functions as well. Its collagen hyaluronate network is responsible for the stability, structure, volume, and transparency and distribution of cells within the vitreous. The cross-rod arrangement of its collagen fibril allows for plastic properties. This is important in distribution of force when the globe is touched or hit, or with movement of the eye. While the hyaluronic acid tangled within the collagen network keeps the fibril from collapsing. The fibrous protein vitrucine framework is what constitutes vitreous collagen. It has fine parallel filaments. It is lowest centrally in the medullary part and is composed of 80% glycine, 8.8% proline, and a small amount of cysteine. Hyaluronic acid is a linear chain of repeating disaccharide units made of N-acetyl D-glucosamine and D-glucuronic acid with small amount of galactosamine. It is responsible for viscosity of the vitreous. It has higher concentration at the posterior cortex, and it has a turnover rate. So as function of the vitreous, aside from structure, we have its shock absorber function. This is due to the viscoelastic properties of the vitreous. It is higher with high hyaluronic acid content, and this protects internal structure of the eye during eye movement, physical activity, and minor trauma. It protects the eye from forces such as friction and vibration. So the vitreous also has flow currents across the retina, between aqueous and between vitreous and lens. The hyaluronic acid in the vitreous is a major resistant to transvitreal flow of water across the vitreous cortex. So flow comes in several mechanisms by diffusion, hydrostatic pressure, osmotic pressure, convection, and active transport. The vitreous also functions as a molecular sieve. Hyaluronic acid interspersed within the collagen network is negatively charged. This inhibits movement of water, large molecule, and cells. This is the barrier to inflammatory proteins. So hyaluronic acid inhibits lymphocyte stimulation, macrophage phagocytosis, and macrophage prostaglandin synthesis. The vitreous is also a metabolic repository. It can be a source of chloride, glucose, galactose, mannose, fructose, hyaluronic acid, and amino acids. It also acts as a sink for metabolic waste, or lactic acid, carbon dioxide, from posterior aqueous, the lens, and the retina. It also has a scavenger function, with the vitreous ascorbic acid as a scavenger for metabolic waste of the lens and retinal metabolism. Now, the vitreous allows for optical clarity, for effective transmission of light from the front all the way to the retina. As I mentioned previously, it has similar index of refraction from the aqueous, so there is not much light bending within. Allows 90% of light between 300 and 1,409 nanometers to pass through. It has a high water content, has fine structure of collagen networks and hyaluronic acid. The medullary vitreous is essentially cell-free and free of macromolecules. So the vitreous can degenerate normally with age. Trauma, inflammation, infection, or myopia can hasten this change. With collapse of the collagen framework, fluid-filled spaces are formed. This gives for some people, this gives the sensation of seeing floaters. Now, with continued degeneration, the vitreous shrinks, causing it to detach from the retina. So, posterior vitreous detachment is basically depolymerization and precipitation of hyaluronic acids, which then leads to pockets of liquefaction, lacunae formation, which later on coalesce. Collagen fibrils become matted and form prominent strands. Vitreous contracts and shrinks. And the vitreous cortex thins out and ruptures, causing this intralacunar fluid to enter the subhyaloid space, separating the vitreous body from the retina. So as was mentioned, posterior vitreous detachment is associated with age and can be accelerated by trauma, intraocular inflammation, presence of vitreous hemorrhage, heat, myopia, and eye surgery. So this picture shows the process of vitreous detachment. The last one to detach is in the area of the optic nerve head. And this is what I previously mentioned as the original connection of in the optic nerve head called the Weiss ring. So posterior vitreous detachment is a natural phenomenon. However, it may pathologically affect the retina and lead to certain disease condition. Because the vitreous is firmly attached at the vitreous base, a vitreous detachment can cause a peripheral retinal tear, which can later lead to a rhegmatogenous retinal detachment if not recognized early. In rhegmatogenous retinal detachment, the liquefied vitreous enters the subretinal space through the retinal break, causing separation of the neurosensory retina from the retinal pigment epithelium. With strong attachment or adhesion in the macula, a posterior vitreous detachment may cause formation of a macular hole. And in vitreous detachment, it can also cause traction on retinal vessel and lead to vitreous or preretinal hemorrhages. So to conclude, we have just discussed how light is transformed into an electrical signal, eventually to a neuronal signal for transport to the brain. The role of the different cells in the retina was discussed. The role of the vitreous in vision and in keeping the structure integrity of the eye was discussed. And we've also discussed how changes in the vitreous can affect the retina. Thank you.